Welcome to the webinar, ADME in Cell and Gene Therapy. Before I introduce Chris Bull, our speaker on today's webinar, I'd like to take a few minutes to tell you about BioIVT. Founded in 1981, BioIVT has grown to be the largest provider of biospecimens and research services to the pharmaceutical and diagnostics industry. We are dedicated to providing the cells and tissues our customers need to help them advance their discovery, preclinical, and drug development programs. We have grown both organically and through strategic acquisitions that have enabled us to develop a comprehensive product portfolio and be the preferred provider for our global biopharmaceutical clients. We have invested in sourcing products globally and an extensive global distribution. We stage products in regions where our customers are located. This helps us to ensure short turnaround times and enable us to get products to our customers' labs when they need them. Underpinning all of what we do is our commitment to ethical sourcing, quality, and a sincere desire to work as a partner with the research community. Here we see a snapshot of the array of BioIVT's product portfolio, including products used in cell and gene therapy programs, normal and disease state biospecimens, and products and services to evaluate the ADME properties of new drugs. We have developed an extensive infrastructure to collect and produce human and animal-derived cellular starting materials that are used in cell and gene therapy programs. Our cell and gene enabling solutions are supported by an IRB-approved, expansive global network of highly characterized and recallable donors. We have over 10 collection facilities across the US and within Europe, and a large immune cell collection database that includes normal, healthy, and disease indications to support downstream development of advanced therapies. All immune cell and ancillary cell and tissue products have robust QA/QC backed redundancy and fresh and frozen formats, providing more flexibility in the supply chain to mitigate risks to your cell and gene therapy pipeline from R&D through clinical development. Our ADME group has the industry's largest portfolio of hepatocytes, subcellular fractions, and recombinant enzymes, as well as other products to support ADME tox and DMPK research. Our team of experts routinely works with clients to help them identify hepatocyte lots with the characteristics that meet their specific requirements. Additionally, we help clients design and implement their preclinical ADME research programs and develop data packages for IND submissions. Our fit-for-purpose study designs include programs in drug metabolism, enzyme induction and inhibition, drug transporters, and other ADME tox research services. Visit bioivd.com to find the products you need for your research programs. Chris earned his PhD at the University of Nebraska and joined Xenotech in 2014 as a research scientist. He held a number of positions at Xenotech that enabled him to gain a deep understanding of the hepatic products used in ADME research. In 2022, when Xenotech joined BioIVT, Chris assumed his current position of Senior Manager of Technical Support. In this role, he is part of our team that assists clients in their research programs. He is available to provide assistance on selecting the right products for your programs, advising on culturing cells and study design, and other technical assistance. When you registered for the webinar, we asked you to respond to questions about off-target effects of the therapies you are working on. I think the key takeaway from these poll results is that researchers are concerned about this, but for the most part have not yet incorporated evaluation of off-target effects into their in vitro programs. Chris, we look forward to your insights about evaluation of tox in cell and gene therapy programs. Over to you.
Thank you, Graham, for that kind introduction. Welcome to the webinar, and today I'm going to give a review on common cell and gene therapy technologies and how people are using conventional ADME test systems to kind of answer questions in the cell and gene therapy technology development. From this point on, I'll start to use the acronym CGT to refer to cell and gene therapy. And we've gained this knowledge through BioIVT working with various CGT companies as part of our relationship as a product supplier. And due to these interactions, we've come to think about how traditional ADME products are used in the CGT field and how they're similar but a little bit different to how people might use these for small molecule development. So for the scientists that have a background in small molecule development, the test systems here listed are very familiar. Primary hepatocytes are the gold standard for ADME work and come in many different formats. Primary hepatocytes can be obtained as freshly isolated cells, cryopreserved, attaching or suspension formats, and as individual donors or pools from multiple donors. And of course, all of these small animal model species can be available. Cryopreserved hepatocytes are not as hard to get as they were in the past, and the quality and the ease of use, culturability, and lot selections have jumped leaps and bounds since the first days of cryopreservation and the offering of cryopreserved hepatocytes. The use of cryopreserved hepatocytes has been well qualified and are commonly used for stability assays, MET-ID, CYP induction, CYP inhibition, among various things, and they're commonly used for hepatotox assays. As we have started working with more CGT companies, it has come to our attention that these groups are using hepatocytes in ways that we weren't used to, and they're using these to develop and de-risk CGT technologies, which I will go into more detail later in the presentation. This slide is a little bit old now, but it shows the FDA-approved CGT treatments on the market and the growth in the CGT therapies. I want to point out that just last year, the first CRISPR-based therapy was approved for use by patients with sickle cell anemia. When this particular technology was first discovered, scientists instantly knew how powerful this was for research and treatment of diseases. A lot of development has been done to get this technology approved, but there's still lots of unknowns around CGT therapies and long-term effects. But these are questions that we need to ask, and we need to put our best efforts into answering so that we can treat patients without putting them under unnecessary risks. So let's talk about CGT, and to do that, we need to define it. So at its most basic, there are 4 types of cell and gene therapy, and some technologies combine different therapy types, making for diverse technologies and very diverse molecular mechanisms. Cell therapies take cells from the patient or donor to enrich for cells with certain attributes or phenotypes. The desired cell population is isolated, expanded ex vivo, and once it's at a high enough density, they're put back into the patient in hopes that the enrichment of these circulating cells will attack the disease within the body and offer relief against the disease or even offer a cure. Gene-modified cell therapies is a combination of 2 different technologies, and these are treatments like CAR-T. This is where patient cells are collected, genetically modified to express certain receptors that will then home in on and interact with the disease cells when placed back into the patient. This will then signal the host immune system to attack the disease tissue and hopefully cure or lessen the disease of the burden of the disease. Gene therapies are generally categorized as in vivo treatments, where cells are not extracted and modified, but the modifications occur in the patients. The gene expression modifying technology is given to the patient in vivo and in order to change gene expression in cellular phenotypes. These therapies use a wide variety of delivery methods, and the goal of these delivery methods is to target specific cells or tissues within the patient to enact a phenotype change. Lastly, the tissue engineered products. These are cells or active molecules that are engineered to restore function or replace damaged tissue or organs. Islet cell transplantation is a good example of this type of treatment, as well as stem cell derived therapies. Now on the right side of the slide, there's a list of diverse products used in cell and gene therapy treatments, and I won't go through all of them, but for simplicity, these technologies can more or less be grouped into two categories. Gene therapies are those technologies that utilize the transfer of genetic material to alter the phenotypes of cells or tissues. And the cell therapies utilize the transfer of cells into the patient that have already have the desired phenotype. So the majority of my talk will focus on the gene therapy side of things. So let's define the more common gene therapy technologies that are being developed in the field. Oligogene therapy, or OGT for short, is one of the hot technologies that many groups are developing and have been shown to be effective. Some people will debate how to classify oligos, and I think most would consider them a large molecule or a biologic, but quite often OGTs are grouped within CGTs, and the debate really stems from the fact that the regulatory agencies still aren't quite sure on how to classify them and what safety tests are needed for these modalities. They will typically fall back on what they know, and they treat the oligos like small molecules and require the typical small molecule safety parameters to be checked prior to clinical trials. OGTs consist of antisense oligos, or ASOs for short. They can be siRNAs, mRNAs, microRNAs, or they use viral vectors that contain DNA that encodes for the various types of RNAs. But the important thing to remember is that at the end of the day, all these technologies' goals is to alter cellular protein expression, to change cellular phenotypes, and this is generally done by upregulating or downregulation of mRNA expression. Another gene therapy that is being widely developed is focused on DNA modifications to the target cell. This can include introduction of exogenous DNA that overexpresses the gene of interest, and this type of therapy is referred to as gene replacement. This DNA can be integrated into the cell's genome and permanently become part of the genome, where it is replicated and passed on to daughter cells when the cells divide. Or the DNA can stay extrachromosomal and is not replicated when the cell divides over time. The extrachromosomal DNA does not have the risk of uncontrolled integration like the integrated DNA does. However, the treatment and expression becomes diluted over time and may require additional doses. Other types of technology is the CRISPR system or some derivative of it. This technology takes advantage of endogenous DNA repair mechanisms, gene sequence specificity, and can be used to knock in or knock out specific genes, or even go in and edit a single nucleotide to fix a mutant protein or coding region. But just like the oligogene therapies, the goal is to alter cellular protein expression in order to alter the target cell's phenotype. These are amazing technologies, but how are these therapies targeted to the right cell or right tissues? With the exception of GalNAc ASOs, most of these technologies require a delivery system to reach the target cells. The delivery systems need to be cell-specific, and they are generally grouped into two categories. You have various viral vectors on the left and non-viral nanoparticles on the right. Both delivery systems encapsulate, protect, and target certain tissues and cells. Viral vectors utilize viral proteins to protect and direct cell tropism, and scientists have developed vectors where they have gutted the viral genome, making replication incompetent, non-pathogenic, but leave behind the proteins that can deliver the therapy. The most common viral vectors that we come across is adenovirus or adeno-associated virus viral vectors. These tend to be chosen because the vast majority of the DNA remains extrachromosomal and offers a good cell tropism. But there's also a concern that a small percentage of the DNA will end up integrating into the target cell's genome. It's pretty rare, but it does happen. Lentiviral or retroviral-based vectors take advantage of the viral reverse transcriptase and integrase proteins to insert the DNA into the cell's genome. This permanently becomes part of the target cell's genome and is replicated along with the target cell's DNA. This potentially makes these technologies lifelong treatments, but each delivery system has their strengths and weaknesses. Nanoparticles are the other common delivery system being developed and come in many different flavors, but it seems that the popular approach is to utilize a lipid-based nanoparticle, or LNP. These have been shown to be fairly tissue-specific and proven in the clinic. Liposomes, micelles, and even loading up cellular exosomes are also being explored. Inorganic nanoparticles made from silicone, gold, or polymers are also being explored. Like all technologies, though, each has its own advantages and disadvantages. For example, the effects of AAV vectors tend to be diluted over time. However, gene expression can last many months, depending on the cell type and route of delivery. Retroviral vectors potentially offer a lifelong single-dose treatment, but uncontrolled integration could lead to, and have led to, unintended gene expression changes in the vector if the vector integrates into a functional gene, or it happens to integrate near an oncogene or activates unintended gene expression. The worst case scenario in this instance is that you get integration in a way that could lead to the development of a completely different disease, such as cancer, especially if the vector were to integrate or disrupt a tumor suppressor, an oncogene, or pro-death signaling pathways. Nanoparticles might be indiscriminately taken up by unintended cell types. This is always a concern, as even though LNPs can be fairly specific, they're not 100% cell tropic. And they're not fully restricted in their cell or tissue tropism. For instance, many people have concerns about different nanoparticles being taken up and modifying germ cells. So even though some of the CGT technologies are approved for treatment, there's still a lot of work to be done to prove that prove to the general public that it's safe, that it's a safe therapy to take. And some of these challenges for the gene therapy research really can be broken down into some of these main topics here. Improving cell tropism or tissue targeting get to the heart of how specific and efficient delivery systems are. Bioavailability is basically stability of the technology in the body. Not only do you want the therapy to be stable while it circulates, you will also want to minimize the immune response in the patient, as you might initiate uncontrolled cytokine secretions, or circulating antibodies might cross-react with the delivery method and render it inert. But what we want to focus on today is talking about non-specific uptake of CGT treatments and potential off-target toxic effects that may come along with it. Typical CGT therapies are intended to go systemic in the body and home to the tissues that are being intended to treat. But the potential problem is that if the treatment goes systemic, it will likely end up coming into contact with highly vascularized tissues. Basically, the concern is the liver, the kidneys, and perhaps the lungs. These tissues offer lots of opportunity for non-specific uptake of the treatment and unintended phenotype changes within those tissues. It is the consequence of these off-target effects that need to be better understood, and we need to answer these to ultimately be able to convince the public that these therapies are safe. And, of course, we need ways to observe if changes made to the technology improve or worsen the efficacy. As we work with various customers, we have noticed that many of the scientists in the CGT field are using many of the conventional ADME test systems to address these challenges. Labs are using traditional subcellular fractions such as liver homogenates, S9, microsomes, lysosomes, and/or tritosomes to assess the stability of therapies while they're in contact with these vascularized tissues. Biofluids such as serum, plasma, and whole blood can be used to give clues to the system, the technology's stability while in circulation. These assays tend to give clues on how long one might expect a therapy to be in circulation before it starts to degrade. We've also found that plated hepatocytes can be and are being used to assess the efficacy of the gene therapy and how modifications of various components might increase or decrease the efficacy of the desired phenotype changes. Human hepatocytes and non-human hepatocytes seem to be the most common use species among them. However, there are groups that are using rodent hepatocytes. And while some groups are using plated hepatocytes to assess efficiency of their technologies, there are also some using them to assess potential off-target hepatotoxicity. While single donor monocultures of hepatocytes might give clues to acute off-target effects of the therapy, long-term co-cultures are used to assess more chronic exposure, such as hepatopac or hepatoimmune. So this slide is intended to give you an idea of how various groups are using hepatocytes to test and develop various components of their therapies. Single donor monocultures are commonly being cultured for anywhere between 3 to 10 days. And some of the delivery vehicle groups are using hepatocytes to assess entry and how, how efficiently they get in. And they're using various molecular methods for these assays. Other groups are using the hepatocytes to assess the efficiency of the gene therapy. This is being done by measuring how many edits have occurred, changes in mRNA expression, or looking for the phenotype changes in the specific cellular protein expression profiles. These assays make a lot of sense to use if they're developing treatments for a liver indication or disease, but we're also finding that groups that don't have a liver disease focus are using hepatocytes and finding them useful. And this is to get to clues about potential off-target liver effects. You can use a standard monoculture for your typical hepatotox assays, looking at acute changes in the hepatocytes. You can use you can monitor LDH, ATP levels, mitochondrial stress, your typical hepatotox assays. But being able to offer technology that can culture for up to 4 weeks allows scientists to look for more chronic effects of the treatment. And this is where a hepatopac may be useful for your program. This is BioIVT's long-term micropatterning cold culture system. And because these cultures can be taken out to 4 weeks, this allows scientists to monitor changes in the hepatocytes in both acute and chronic exposure settings. You can do the typical hepatotox readouts, or you can use more advanced molecular techniques and look at changes in gene expression over time after exposing the cells. What you might find is that some treatments are more acutely toxic to the hepatocytes. Some may take longer to show the tox phenotype. Or it could be that the cells become stressed, but can eventually recover. Exploring these changes in the hepatocytes might give clues to what might be expected in the small animal models or once it goes into the clinic. As a biologist, I find this really interesting, and what I think other people should be interested in trying to understand is how the modified cells are going to compensate for the different treatments. Scientists should be thinking about how those phenotype changes in the cells and tissues will affect the fitness of the modified cells. Any changes that you make inside those cells are going to change how fit they are in relation to the greater system. Will treated cells be more fit in the context of untreated tissue? Are those cells going to eventually take over and repopulate the liver? Are they going to become less fit? And maybe they'll die off and you won't have to worry about off-target effects, as long as they don't die off all at once. Will those changes lead to dysregulation of cell signaling pathways or upregulation of oncogene or tumorigenic pathways? These are all vitally important questions that need to be investigated, especially as scientists start to look to use these technologies to treat non-lethal, non-monogenetic diseases or diseases that have effective treatments already on the market. While these in vitro test systems cannot predict everything, I believe that they can give clues as to what to keep an eye out for in the small animal models or in the clinic. These treatments demand long-term patient follow-up to fully understand the long-term effects. But as treatments evolve and develop further, the ultimate questions and concerns don't really change. And the more clues we have can help groups de-risk their programs and develop and perhaps deprioritize certain programs before they go to the clinic. Groups that are using viral vectors have very similar questions as those that are using other delivery systems, but they also have the added concern that the vector will cause an immune response. It could be pre-existing cross-reactive antibodies, or it could be a de novo innate or adaptive immune response. For these groups, we feel hepatoimmune might be more useful for them. This is the hepatopac system that adds in Kupffer cells to create a long-term micropatterned triculture and adds in the liver's resident macrophage into the equation. Hepatoimmune can be supplemented with serum, HLA-matched Kupffer cells, and in the future, we're looking at being able to add other cell types that might be useful for assessing if there might be an immune component to potential hepatotox phenotypes. You would essentially treat the triculture just like you would with hepatopac. You do all the same assays, but with this system, you could add in a cytokine release endpoint. You could look at how the treatment activates and maybe leads to cytokine release from the Kupffer cells, or you might find that the cytokine response actually starts with the hepatocytes themselves and then involve the Kupffer cells or other immune cells as the hepatocytes respond to the treatment. These aspects of the treatment might be important for groups using viral vectors, as many of the deaths in CGT clinical trials involve an immune response component. Furthermore, various secreted cytokines are known to affect hepatocyte function, specifically inhibition of various P450 enzymes. And given that many of these patients will likely be taking co-administered drugs, these assays can give clues to potential drug-drug interactions that may need to be addressed. So we have described how conventional in vitro ADME test systems can be useful for CGT development. And I think that these types of assays will become more important as programs look to treat larger populations for multi-genetic diseases that can perhaps be controlled with various medications and lifestyle changes. As we have worked with various customers in the CGT realm, groups have more, the ADME and the CGT groups have more in common, than we first realized. And they're ultimately asking very similar questions, just using slightly modified methods. So this is an area of research that BioIVT can support along with our CGT business unit. We have a lot of information can be found on our website. It's bioivt.com. We also have a sister website, which is xenotech.com. Much of this information is being slowly migrated over to bioivt.com. And once that occurs, the Xenotech website will no longer be used. But in the meantime, you can still find very useful information on that website. Again, my name is Chris Bohl. My email is below. And my job is basically to provide technical support for your research projects. I can help pick lots that have the characteristics that you're looking for, and we can even come to your lab and teach you how to use these test systems if you're unfamiliar with them or haven't used them much in the past. So thank you for your time. I hope that this has been useful, and hopefully we'll be in contact and I can help you with your research in the future. Thank you.
Thank you, Chris, for a really great presentation. And while we wait for questions to come in, also invite everyone to go to bioivt.com, and you can go to our resources section where you can search for many different kinds of resources, from webinars or videos we've done in the past, posters. There's other educational content, including tech sheets and so on. And there you can also register and subscribe to newsletters and other materials that come from BioIVT.